problems. Of the problems. So part 1, we're given a list of proteins and effector molecules and DNA sequences, and we're trying to determine when they're used. So for part A, part B, small effector molecules are an inhibitor, allolactose, CAMP, and inducer. Sequences that are found in the DNA are the operator, +1, -10, LAC-A, and LAC-Z. So even though the LAC inhibitor is present, it's not actually involved in the LAC operon. But the structural genes are LAC-Z and LAC-A. For part 2, we're filling in the blanks. The LAC operon is regulated by 2 inducers, CAMP, or cyclic AMP, and allolactose. The binding site for the sigma factor and RNA polymerase is the LAC-P, so the promoter. While the CAP site is the binding site for the activator that regulates the LAC operon. Adenyl cyclase converts ATP into CAMP. The 35, 10, and 1 sequences are found within the LAC promoter portion of the LAC operon, so that it can be recognized. The LAC-L gene encodes the LAC repressor, while the LAC-Y gene encodes lactose permease. Attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And the E. coli strain that contains 2 copies of the LAC operon is called the merozygote strain. For part 3, we're doing a little bit more problem solving. We're trying to figure out under which circumstances the cell will turn blue. So for part A, it's not going to turn blue even though glucose is absent and X-gal is present, because IPTG is also needed in order to turn it blue. For part B, we do have IPTG, we don't have glucose, which is good, and X-gal is present, so the cell will turn blue. For part C, we do have IPTG and glucose, but X-gal is needed to be cleaved to turn it blue. So even though it's producing it, we can't tell. For part D, IPTG is present, but so is glucose, and glucose represses the operator, so the cell won't turn blue. There's the same problem for part E, and even if glucose was present, X-gal is absent, so we would have the same problem even though it would be actually functioning. For part 4, we are looking to see if something is constitutive, inducible, or non-inducible for the LAC-Z gene. Inducible means transcription can take place. So for A, all of the pieces are functioning normally, so we know it's inducible and it will take place normally. Non-inducible means something's preventing it from taking place. In this case, it's the IS, because the repressor can't bind to allolactose, and allolactose is what would change it so it could be activated. For part C, it's constitutive. Now, constitutive means that it's going to happen and it just keeps on happening. And in this case, it's because the OC prevents the attachment of the repressor for both the plasmid and the chromosome. For part D, it's non-inducible. It can't be transcribed because, again, the IS on the F plasmid is going to interfere with how things work. Part E, it's constitutive because even though there's an IS on the chromosome, a functioning one on the F plasmid, And even though there's a non-functioning operator on the F plasmid, there's a functioning one on the other. So they kind of just help each other out, and they're always making transcribing. For question 5, we're looking at how yellow liquid turns depending on what's in it and whether we are given it in the presence of lactose or in the absence of lactose. So for the first one, we look at our genotype, and no yellow color is going to be produced because the lac L is non-functioning and the lac P, so transcription can't take place. For part B, everything checks out except the operator, but that's just making it so it can't be repressed. So we're still going to get a concentration of 1 for lactose and non-lactose. For part C, we're going to get 2 grade 2 for the yellow in the presence of lactose, because both of them can transcribe it. But if there isn't lactose, the F plasmid has a F plasmid is the only one that can produce, that can transcribe, because the other one can be turned off because as a functioning operator. In part D, if we have lactose, we can make concentration of 1. And this is because the F plasmid continues to make to transcribe, whereas the chromosome is broken, and so without lactose being there, it doesn't do anything either. For part E, nothing's going to be made for either one because they're really messed up. So the lac L on the chromosome doesn't work, and it also doesn't work on the F plasmid, so transcription can't even take place. I hope that was helpful and clear. Thanks. Bye.